Let's use this particular slide to practice a little bit what we've learned so far about the processes of diffusion along with osmosis. So before we do that, though, let's review, right? So diffusion is going to be the movement of particles down their concentration gradient. And in this particular case, we're going to be looking at diffusion across a selectively permeable membrane. So remember, as particles move across the membrane, or let's say solutes in this case within a solution, move across the membrane, they're going to be moving down their concentration gradient. Also, remember that solute particles, as they move, they're elitist. They only care about their own concentration when they're taking into consideration which way they're going to move across the membrane, right? So each solute will look at its own concentration gradient and only move down its concentration gradient, going from an area where it itself is highly concentrated to an area where its concentration is less, not taking into consideration any other solutes within the solution. Osmosis, on the other hand, right, this is going to be the movement specifically of water molecules across a selectively permeable membrane. And in this case, water likes everybody. It wants to keep everybody happy. So water takes into consideration all the solute particles that are dissolved within the solution before it decides which way it's going to go. Remember, for water is going to go from an area of low solute concentration to an area of high solute concentration to try to sort of dilute out that high solute concentration. All right, so let's take a look at our, let's say, cell, right? So we have a balloon here, which is going to mimic our cell, and the membrane of the cell is going to be this sort of latex balloon, which we filled with a particular solution. In this case, we have a 0.03 molar sucrose and 0.02 molar glucose solution on the inside of our cell. And we've taken our cell and we've put it into a beaker that's filled with an environmental solution. In this case, in the environment, we have 0.01 molar of each sucrose, glucose, and fructose. For the sake of this example and any of the examples on your assignment, sort of assume that there are equal volumes of solution on the inside of the cell as well as in the environment, just for simplicity's sake. All right. So let's say we're looking at osmosis first, and we have this cell with its membrane, and the only thing that can pass across the membrane is going to be water. So first of all, which direction is the water going to go? Is it going to go into the inside of the cell via osmosis, or is it going to leave the cell and go to the environment? So take a minute, pause the video if you have to, think about it. Which way is the water going to go? All right, let's take a look. So if I'm going to draw a little bit on the screen here, so we have our cell, right? And the internal contents of the cell are going to be your sucrose and your glucose. Remember, water cares about everybody, so we have to take into consideration all the solutes inside the cell versus all the solutes in the environment. So to do this, essentially what we could do is we could just add, right, the concentrations of both sucrose and glucose in the cell. That'll give us a total concentration of 0.05 molar. And we'll do the same thing for the environment. So we'll add all these guys up. In this case, we'll end up with 0.03 molar. You can tell now that on the inside of the cell, we have a higher concentration of solutes versus the outside of the cell. So in this case, we are going to have the water chasing high solute concentration. It's going to go from the outside of the cell in to try to dilute out that high solute concentration on the inside. All right, let's try another one. So let's say that the membrane is only permeable to sucrose. That's it. All right, now think about it. So it's only permeable to sucrose. That means that the glucose concentration is not going to change inside, outside the cell. Fructose concentration is also not going to change because they don't have the ability to move across that membrane. Only the sucrose concentration can change, right? So in this case, we take a look, right? We have 0.03 molar sucrose on the inside of the cell. On the outside, we only have 0.01 molar. Clearly, the concentration of sucrose is higher on the inside of the cell than the outside. And so in this case, sucrose is going to have a net diffusion direction from the inside of the cell out to try to balance things out. Once the cell reaches equilibrium with the environment, what is going to be the concentration of that sucrose? Well, let's take a look. We have, let's say, a total of 0.03 molar sucrose on the inside of the cell, 0.01 molar sucrose on the outside of the cell. Assuming equal volumes of these two solutions, what we could do is simply take the two concentrations, add them together. Right, in this case we get 0.04 molar as total, and then we would just split that concentration between the two solutions. So inside the cell we'd end up with 0.02 molar, and then on the outside we would also end up with 0.02 molar sucrose in both cases, and that particular solute has reached equilibrium. And even though the sucrose is still going to move in and out of the cell, it's going to do so at the same rate. So if the same number of molecules that go into the cell, the same number of molecules come out. All right, let's do one more. How about fructose? Let's say fructose is the only thing that can come across that membrane. Which way is it going to go? Well, this one might be a little bit tricky because we actually don't have any fructose on the inside of the cell. So you might not be sure what to do with that. Right, so technically you can consider the inside of the cell as having 0 molar fructose. So again, we're going to do the same thing as we did with the sucrose. We want to balance out the fructose concentration. So in this case, we would take 0 molar fructose on the inside of the cell plus our 0.01 molar fructose on the outside of the cell, add those two together, and we get a 0.01 molar total concentration of fructose. If we divide that by 2, right, to split the amount that we have between the two solutions inside the cell and outside the cell, we would then end up with 0.005 molar fructose inside the cell, and the same 0.005 molar fructose on the outside. All right, so hopefully that will be helpful in determining the answers to some of the questions on your assignment. All right, so before we leave this slide, let's take one step back, right? And remember that we had identified through the process of osmosis, right, we have more solutes on the inside of the cell than the out, and so water is going to chase that high solute concentration. It's going to go into the inside, right, inside here of our cell. And so what's going to happen to that water balloon, right? Is it going to expand and get bigger, or is it going to shrink and get smaller? Think about it, right? So in this case, if water goes into the water balloon or into our cell, it's going to end up increasing the size of the cell. And this is in fact exactly what happens to a variety of cells depending on what kind of solution they're hanging out in. They can gain or they can lose water as a result of being in that particular solution. All right, so this is going to bring us to now a discussion of something called tonicity. So tonicity is the ability of a surrounding solution to cause a cell either to gain or to lose water. Now, tonicity sometimes can be a little bit hard to think about, especially the way that the definition of the different situations for tonicity are listed on the slide. So let's talk about the slide first, and then we'll talk about maybe some caveats. All right, so tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. So for example, if you have a cell and you've placed the cell in an isotonic solution, this means that you have the same solute concentration on the inside of the cell as you do on the outside. So in this case, water is going to move in and out of the cell, but it's going to do so at exactly the same rate, right? So in this case, there's going to be no net water movement across that plasma membrane. The cell's neither going to gain nor lose any water net. Okay, so now we could take the concentration of solutes and change them on either side of the membrane. If we were to take a cell and we were to place it in a hypertonic solution, you can think of hyper like high energy, also having high solutes. A hypertonic solution has higher solutes than the solution on the other side of the membrane. So if we take a cell, we place it in a hypertonic solution, right? In this case, water is going to end up chasing that high solute concentration on the outside of the cell. Water is going to leave the inside of the cell to come out, and the cell is going to shrink and lose water. The opposite is going to happen when you place a cell into a hypotonic solution. So hypo, think like hypothermia where you're cold, the temperature is really low. Hypotonic solutions will have less solute concentration than what is on the other side of the membrane, right? So in this example, we take our cell, we put it into a hypotonic solution. In this case, the cell actually has more solutes than the outside. And so water from the outside of the cell is going to rush into the inside of the cell. The cell is going to end up gaining water. Now, the way that we have described isotonic, hypertonic, and hypotonic has been all sort of in this context of putting a cell into one of these different types of solutions. But if you think about it, when we actually, let's do it this way. When we look at tonicity, we can look at tonicity essentially just in a way, as a way of comparing two solutions on either side of a membrane, right? So we can actually go back to our beaker of water, right? We had our nice little beaker of water with a selectively permeable membrane in between. And if we were to then, let's say, put some solutes in here, we could put a lot of solutes, say on the left-hand side, and only a couple on the right. We can use those same tonicity terms to describe these two different solutions, right? So we could say that the solution here on the left-hand side is hypertonic in comparison to the solution on the right, or you can say that this side is hypotonic in comparison to the solution on the left. All right, so remember, hypo means low solute, and hyper means high solute, right? So in case maybe that hypertonic was invisible, hypertonic on that side. So in this case, it doesn't matter, right, whether what's inside the cell versus what's outside. What I want you to understand is when we're looking at tonicity, we're really comparing two solutions, right, on either side of a selectively permeable membrane. One side might have more solutes, that one will be hypertonic, and the other one might have less solutes, and that one will be hypotonic. All right, so hopefully that helps a little bit with this discussion of tonicity. All right, so this idea of tonicity can actually sometimes cause problems for a cell. And the kinds of problems it can cause might be a little bit different depending on whether we're actually talking about either a plant cell or an animal cell.